Thank you very much. So I'll be presenting today, the activities that we've been conducting in the last, I want to say 2 to 3 months, including some of the data we have accomplished and what we are actually working on right now and working to achieve in the upcoming years or months, I should say. So again, a brief reminder of what Defense Therapeutics is. It's basically a biotech company that is working on a very innovative and highly versatile technology called the Accutox technology, which I'll be discussing in a few minutes. Basically, we're developing the next generation biopharmaceuticals that are targeting multiple indications, whether it's in the field of immuno-oncology for cancer or infectious diseases such as COVID, HPV, and so on. So the platform is really a technology we call Accutox, which stands for the accumulator, because what it does really is that it accumulates any biomolecule of interest into a target cell. So you can think of proteins, RNA molecule, DNA molecules, antibodies, whatever you wish to actually introduce into a cell of interest. And the company is actually very well positioned in terms of the business side because one, we are working with a technology that has multiple applications. We have a very attractive capital structure with only 36 million shares outstanding. And with all the phase 1 trials, we are actually looking forward to start by the end of this year, if not early 2023. I think we are well positioned to achieve data-driven value inflection points based on what we will be achieving in these phase 1 trials. And of course, what makes us also very strong is the experienced team that we were able to assemble together, which is, of course, composed of the CEO, Mr. Plouffe, Joseph Meager, our CFO, who's been taking care of the money and everything related to the financial structure of the company. We have multiple advisors such as Michael Flederer, Riam Shama, Dr. Raymar Lomenberg, and Dr. Sarkis Mitarisian. So these are experts that have a lot of knowledge, whether it's in the clinical development or regulatory affairs or, in general, scientific advisors to help us really move our products forward into the clinic. So what are the real current challenges that we're actually facing when it comes to immuno-oncology and or infectious diseases? Well, we know, for example, in the case of cancer, that so far, the only major treatment we have is what we call standard of care. So I'm talking here about chemotherapy, radiotherapy, or surgeries. Of course, there are some cancers that could be treated with these approaches. I can give you examples, for example, breast cancer at early stage or melanoma at early stage. But the problem is once these tumors end up being disseminated into the body, it becomes practically impossible to treat the patient. So we're talking about stage 3 or stage 4 because the tumors are all over the place and it's very hard to really get access to these tumors and retrieve them in using the standard of care. Now, there are multiple technologies that have been provided by pharmaceutical companies or biotech or biopharmas. But again, these targeted biopharmaceuticals are always struggling with molecular obstacles that are limiting the potential of the drug that is being used. For example, you have resistance that could be developed, you have molecules that are mutating, or you have, in the context of ADCs, the entrapment of the molecules within the endosomal structures, which end up destroying the product or not allowing the product to actually reach its target site. So these are all issues that we have to actually cope for when we develop, the different biopharmaceuticals. Of course, we've been hearing a lot lately about vaccines. If we go to the era before the pandemic, vaccines were never that exciting. They were never that attractive because people said, if I can treat a disease with a drug, why bother with the vaccine? But now we understand the value of vaccines in the sense that they can not only help us protect the population from specific infectious diseases, but on top of that, it can generate also a memory response, which will make sure that the patient or the subject is actually protected for the next 15 to 20 years. And if you talk, for example, in the context of cancer, there is no real cancer vaccine available on the market at the moment. I mean, there's only one product that was approved by the FDA, which I'll be talking about briefly. But even then, it's highly questionable due to the low efficacy or the potency of that vaccine. And of course, we're still missing a lot of vaccines against deadly pathogens. And of course, you have to keep in mind that there will be new pandemics that will be coming in the next 5, 10, 20 years. And so we have to be ready really to deliver effective vaccines against these different pathogens. So there's always an urgent need for an enabling and first-in-class versatile technology that can enable us not only to develop something that is efficient, but something that could be applicable to any condition. If you have one drug targeting one specific molecule or target, you're stuck with this. But if you have a technology that could be applied for infectious disease or pathogen 1, 2, 3, or cancer 1, 2, 3, then you can deliver multiple products no matter what the indication is. So what is really the Acume technology? Well, in simple, it's a very small molecule that is of 2.5 kilodalton, so extremely small, that is really composed of the bile acid fused to a nuclear localization signal. So very simple to synthesize, very simple to produce under GMP condition, GMP manufacturing. We own the technology, so Defense is the sole owner of the technology. There are no universities involved or other institutions beside the investors, of course. And what this molecule does is that it really promotes endosome to cytosol escape. So in order to really show you how this molecule works, here's another example of an experiment we conducted, which visually shows you how this molecule works. So if you take, for example, a cell, a dendritic cell, which is an antigen-presenting cell we have in our body that presents antigens and activate the immune system, you can take a molecule called the enhanced green fluorescent protein, which is a fluorescent molecule that is fused to another molecule called galectin-3. So what galectin-3 does is that it binds to carbohydrates, so proteins or molecules that have sugar residues on them. And these molecules are usually found on the extracellular space, on the membrane of the cell, if not within the endosome, because the endosome is just an invagination of the cell membrane, which is really composed of the same component that were on the cell membrane initially. Or you can find those carbohydrate residues in the Golgi, which is not something we're going to be talking about today. So if you express that molecule in a dendritic cell, the entire cell is green because the molecule is expressed in the cytoplasm and it's not really localized or binding to something specific. Now, if you add the Acume molecule, what the Acume molecule will do is that it will break down physically the endosomal membranes. So what that means is that now this human galectin-3 fused to GFP is going to be able to migrate from the cytoplasm and localize in these endosomal structures, because now it can see the proteins inside these endosomes. And as you can see now, you're going from a cell that is completely green to a cell where you can see puncti or roundish structures that are green, which are broken endosomes. So this is really what the Acume can do. And you can imagine if you have, for example, an antigen, a protein, an ADC, that will be able to then move to the nucleus, to the cytoplasm and or the nucleus where it can hit its target. So what are the various applications of the Acume technology? You can apply it into antibody drug conjugates. You can apply it into vaccines. If you have, for example, an antigen you wish to use as a vaccine, I'll give you an example in a moment. And of course, the applications are endless. If you have an RNA molecule you'd like to develop or deliver, that's the way to do it. If you have, for example, if you want to optimize your CRISPR delivery mechanisms, we have data on that as well. So the applications are endless. And the other thing is the company owns all the IPs. So we are completely independent in that regard, and we're moving our technologies forward. So this is really a table that shows you overall what are the major programs we are actually working on right now. So we have a dendritic cell vaccine, which we call the Accuvac D001. And this is a dendritic cell, it's a cell vaccine that is targeted for melanoma. And if it does work in melanoma, we could then apply it in different indications such as breast cancer, lymphoma, glioblastoma, colon cancer, and so on. We have also developed a protein-based vaccine with the Acume molecule, which is targeting cervical cancer, because that's another cancer that we have to be aware of in terms of what happens if the Gardasil 9 doesn't work, or you have new variants of the HPV or that are actually appearing, which is happening right now in Asia. So that's something that we have to actually focus on. And I'll talk also about the Accutox, which is a modified Acume molecule that could be used on itself as an anti-cancer molecule. And so that's something we would like to aim or to use against melanoma and breast cancer. And of course, we have an ADC program that is quite advanced and developed, but right now we're waiting for the data coming from Europe, from Finland and France, early summer, so June, July. So I won't be able to present data on that program at the moment, but be aware that we're going to have amazing results on antibodies that are actually being tested as we speak against breast cancer and PDX models. And we have also plenty of data on preclinical data on the COVID vaccine, and that's a program we're also moving on into phase 1 trials eventually. So today I'll be focusing on the DC vaccine, on the protein-based vaccine, and of course on the Accutox program. So as I mentioned initially, there is only one FDA-approved vaccine against cancer, and that's Sipuleucel T. It's actually developed or manufactured by Dendreon in the States. And that's a vaccine where you can take a dendritic cell from a patient. You then treat that dendritic cell with a PAP protein, which is an antigen expressed on these on cancer cells, fused to the cytokine GM-CSF. And so the idea there is that the monocyte is going to uptake the antigen, get the signal to start differentiating and maturing into a dendritic cell. And then you can take those dendritic cells that have now the antigen, infuse them back into the patient so that they can activate both a CD4 and a CD8 T cell response, which then would lead to an attack against prostate cancer. So that's all amazing. But unfortunately, if you look at the data that was actually generated in patients, the vaccine didn't deliver the hoped for outcome in the sense that patients were always, surviving 4 to 6 months more than placebo or control group. Now, I know that, they were advanced on their time. At that time, we didn't have, in the past, when the vaccine was developed, they didn't have the immune checkpoint blockers and all the different knowledge we had on DC biology. But that's what the vaccine of Sipuleucel T can deliver at the moment. So there is an urgent need right there because there's no real other cancer vaccine alternative to the one that is being manufactured by Dendreon. So we came up with a study where we demonstrated that if you can basically take the Aq molecule, link it to proteins or protein lysate or antigens, and then add it on the DCs, you can dramatically enhance the T cell response. And here we're talking about an enhancement that goes from 50 to 100 folds, which is significantly amazing if you think about it from that perspective. So this study was published in a prestigious journal, the Cell Reports Medicine. I encourage you to go have access to the PDF file, which explains all the data all the experiments we've conducted. But the highlights of the study is that Aq-linked antigen actually enhances the processing and the antigen presentation process. It leads to potent T cell response, which I'll be covering in a moment. And when you apply it to allogeneic DCs, meaning DCs that are coming from non-related patients or patients that have, subjects that have nothing to do with the patients. So they only have non-compatible dendritic cells, which we know can enhance significantly the pro-inflammatory response. It acts like an adjuvant, the natural adjuvant to the immune response. That even works better. And what's really interesting is that we were able to demonstrate within the tumor that we can increase the CD8 to Treg ratio, which is a very major achievement if you think about, the tumor microenvironment and the immune infiltrates. So how did we actually test the vaccine? This is one of the studies that that we have completed in our study using the allogeneic dendritic cell vaccine. So what we did is we first planted the tumor at day 0, and then we delivered the vaccine 2 different doses at 3 times 10 to the 5 dendritic cells. Of course, they were pulsed with the lysate and the Aq molecule. And then we delivered the immune checkpoint blockers, the anti-PD-1, just one example, at a specific dose that has been used ultimately in preclinical research. And then we looked at 2 things. We looked at tumor volume over time, and we looked at the survival of these animals. Have they reached the endpoints? And as you can see in panel B, if you look, for example, at the red line here, this is the group that received the allogeneic dendritic cell vaccine containing the Aq antigen plus the immune checkpoint blocker. And in this case, we got 3 out of 10 animals that had a complete response, meaning the tumors were actually shrinking and disappeared. And we have 4 out of 10 animals that got a partial response, meaning that the tumor was not growing or shrinking. So it reached a stage of equilibrium where the immune system and the tumor were fighting and not allowing the tumor to actually grow. And that really translates into a 70% survival over time. Now, 70% survival using a dendritic cell vaccination approach is something big. The reason I'm saying that is that if you look, for example, at a European company right now, which I won't mention, they're publicly traded at the moment, and they have a DC product, an allogeneic DC product that they use in combination with the immune checkpoint blocker. What they can reach basically is just a delay in survival, no real cure, no survival beyond even 20 or 30%. There are plenty of other examples where people have been able to reach 20 or 30% in survival. So that's a major achievement in our hands. And if you look a little bit more into the mechanistic, so what's happening exactly in the tumor itself. So what we did in this case is that we vaccinated the animals, we collected the tumors, and then we digested them to understand what was happening inside of them in terms of immune infiltrates. So then we looked at a variety of different immune cell markers, CD11B and CD11C. Basically, these are markers of myeloid cells, so macrophages, neutrophils, dendritic cells, anything that is myeloid. And what was really intriguing was the enhancement in the DC vaccine that contained the Accutox lysate and the PD-1 in the recruitment of CD11C positive cells, which are mostly dendritic cells. So that means our allogeneic product was recruiting endogenous DCs, which are able to uptake or capture the antigen that we're delivering from our cells and presented in parallel to responding T cells. So that's really, it's like if you have a booster happening in the tumor itself. We looked at the CD4s, we looked at the CD8s, and we saw a very nice improvement in CD8 infiltration along with NK cells. So NK cells are the natural killer cells. These are cells that have a specific job to look or screen in an MHC independent manner for cells that are either virally infected or being transformed into a cancer cell, and the recruitment enhancement means the tumor could be controlled and eradicated. And we also looked at CD19, which is a marker for B cell responses, or B cells in general. So we were really intrigued by this recruitment of those immune cells. And when you looked at the regulatory T cells, so these are your biggest enemies when it comes to cancer immunotherapy, because these are the T cells that will come in to dampen the inflammatory response. So cancer cells love them because once they are recruited to the tumor, they secrete TGF-beta, IL-10, and so on to be able to really inhibit inflammation. And what we saw, it was a decrease in the recruitment of these regulatory T cells. And when you do the ratio of CD8 to Treg, you can see that our vaccination was able to enhance the recruitment of CD8 at the expense of Tregs. So that's really the hoped for outcome you would like to see in a tumor, which means that you are able to transform a tumor from cold to hot type thing. So this is a slide here that summarizes really what our allogeneic DC vaccine does. So you inject the vaccine, you promote inflammation. We've got a lot of other studies about cytokines and chemokines being produced. That recruits a variety of different endogenous immune cells, such as CD8 and NK cells. That activation also recruits the endogenous dendritic cells, which could be cross-primed and help in promoting a CD8 T cell response down the line. So with this being said, we published these data. We actually established a contact with a group in the UK to start our phase 1 trial using a dose escalation study, a 3+3 design, which is planned for Q1 of 2023. So right now, what is happening is that we are basically conducting the so-called dry runs. So we're just making sure that the manufacturing process is actually going very well, so we can present it to MHRA and eventually get the approval for a phase 1. So this is done in collaboration with the Birmingham University, where the DC manufacturing site is located. So that's a clean room. And the trial itself will be done at the Christie Hospital in Manchester. That's where the patients will be receiving the therapy. So I'm going to switch gears and talk about another cancer vaccine, but now it's a protein-based vaccine targeting cervical cancer. So you all probably know that HPV is the infectious agent responsible for triggering cervical cancer. That's really one of the few infectious agents that we know can cause cancer. Now, active development is being conducted, or research is being conducted to understand if this is related to liver cancer and whatnot. But HPV is for sure established as the agent driving the development of cervical cancer. And as you probably know, Merck and GSK have developed very effective vaccines against the viral particles themselves. Initially, Merck developed Gardasil. Now they brought in Gardasil 9. And the reason why they added a 9 after that is because they're basically mixing 9 L1 proteins or capsid proteins derived from different subtypes of the HPV. So subtypes meaning different strains of the HPV that could be causing cervical cancer. The only problem is the vaccine is not effective against the other subtypes that are remaining there, and there might be new subtypes that could appear. And what's really interesting is that there are apparently some new subtypes that are causing right now cervical cancer in Asia. So there is still room for improvement in that context. But the other thing I'd like to mention is that this vaccine is only used prophylactically, meaning you can inject it to patient to protect from the infection itself. So if the infection happens and the patient develops cervical cancer, it's too late because the vaccine is really targeting the viral particle, not targeting the cancer itself. So it couldn't be used as a therapeutic vaccine in the sense that post-infection or once cervical cancer has appeared. So how does HPV develops or trigger cervical cancer? So the good thing about today's technology is that we can sequence all kinds of viral particles, the genome, we can understand, the genes that are being expressed and so on. And that's why we were able to respond quite fast to COVID. And if you look at the genome of HPV, it's composed of various different genes. So of course, the L1 protein, these are the capsid proteins that Merck and GSK are using for their vaccine development. But you've got other genes that are involved in growth stimulation, replication, transcription, and whatnot. And the two genes that are quite important for cervical cancer are the oncogenes E6 and E7. And the reason why these two are quite important is that these are the ones that play a very important role in the transformation process in the epithelial cells. So what the HPV E6 oncogene does is that it blocks the proapoptotic protein P53, which, as you know, is the master regulator of apoptosis. So that's the protein that can tell to the cell, you gotta die, otherwise you'll become a cancer cell. So by blocking P53, you block the cell from dying, and that's a sign that the cell is being transformed. And the HPV E7 protein is also a protein that can target P53, but it can also target multiple other proteins involved in growth or growth arrest, like P21 or the RBE2F factor. And therefore, if you would like to treat cervical cancer, you really have to block the activity of one of these or these two proteins simultaneously. Otherwise, the cell will completely lose its ability to induce cell death or kill itself by apoptosis. So this is where we said, okay, well, that's an opportunity for us to apply the Accum technology against cervical cancer. So we took the E6 and the E7 protein, we added the Accum on there, we delivered the vaccine as 2 doses at day 0 and day 14. And 2 weeks later, we gave a challenge with the C343 cell line. So this is a cervical cancer cell line for preclinical studies. And there what we noticed is that the group that was completely protected with absolutely no tumor appearing was the Accuvac PT007, which is basically the E7 protein to which we added Accum. Now we have some preliminary evidence demonstrating that one of the reasons also why this might have been improved for the E7 is because we have changed with the introduction of the Accum somehow the structure of the protein, making it more stable, and also changing the charge of the protein, which makes it way more easier to uptake it by antigen presenting cells. And so as you can see, the animals were completely protected. These animals were then challenged not only twice, but 3 times. So the initial challenge was at day 28. Then we gave a challenge about a month after, and then we gave another challenge about another month after, for a total of 3 challenges. And that experiment is still ongoing. So we're gonna keep it on until day 96, which in the lifespan of a mouse is a long time, because 1 day in a mouse lifespan is about 48 days in humans. And of course, all of these animals are, surviving up to now. So that's quite interesting. That's a prophylactic vaccine. So now we can change, for example, the Gardasil 9, which is composed of 9 proteins, into 1 single protein that could protect the subject from cervical cancer itself. But can we actually use it as a therapeutic vaccine? In other words, can I give it to patients that have already contracted the virus and developed cervical cancer? So what we did in this case is that we transplanted the tumor cell line first, and then we delivered the vaccine a couple of days after when the tumors were palpable, along with a commercially available GMP or FDA approved adjuvant. And then we delivered antibodies along. So we delivered immune checkpoints with the vaccine. And what we noticed in case is that, yes, the vaccine could actually control tumor growth. If you deliver it with, we delivered it with anti-PD-1, anti-CTLA-4, which are common immune checkpoint blockers being tested or being approved for use against cancer in patients. We tested it along with the 4-1BB antibody, which is an agonist antibody activating T cells. And of course, we tested it against the latest immune checkpoint that is practically very hot in the market right now, the CD47, which is an immune checkpoint that blocks the "don't eat me" signal, so allowing the phagocytes to really attack tumor cells. And as you can see in this case, the group that did very well was basically the Accuvac PT007 in combination with CD47. We do still see a good response with the other immune checkpoint blockers, but the CD47 seems to be the one that drives the best response. That being said, this experiment was done with one single dose of the dosing, in the sense one single concentration of the vaccine. So right now, we're trying to see whether increasing the dose or the concentration of the protein could actually enhance significantly the response to be able to reach, for example, 70, 80, or even 100% protection or survival on the long run. So right now we are also in discussion for a phase 1 trial, although this phase 1 trial, we would like to conduct it in North America, in Canada, or the US. So we're still in discussion for the site where it's going to be held. But that's something that we are working on as we speak, and we should be able to actually get a clearer idea in the next month or so. So the last indication I'll be talking about today is the Accutox program. So this is the use of the Accu molecule on its own directly as an anti-cancer molecule. So the reason we said multiple indications, because we have evidence that this molecule could actually be used against any kind of cancer type, and we've ourselves tested 3 of these. So we're hoping eventually to be able to test it against a variety of other cancer indications. So how was the Accutox program born? So initially, when we conducted the studies, we were basically linking Accume to the antigen. So that's a chemical manufacturing process. It was added on resting DCs, and then the DCs were activated, and that's what we've done in our first study. But then we asked the question, I mean, do we really have to link Accume to the antigen through chemical reaction? Can we just take it and mix it together with the antigen and just dump it on the dendritic cells? And the answer is no. If you do that, the dendritic cells will end up dying, and that's exactly what we found. So that brought us to the point that, oh, maybe this molecule is actually toxic, and maybe we can exploit it as an anti-cancer molecule. So initially, when we conducted an IC50 analysis, so that's an analysis where we take different concentrations of the Accu molecule, we add it on cancer cells. In this case, it was done in conjunction with the EL4 T-cell lymphoma. We noticed that, the IC50 was 95 micromolar. So these are all the group controls. So the bile acid alone, the nuclear localization signal alone, or the vehicle alone. And in this case, we're not able to kill the cells. It's only the Accume itself that was able to really kill the tumor cells with time. However, we were not really happy with the IC50. We were looking for something way more powerful than the original Accume. So our CTSO in the company designed a library of over 100 different molecules. So we had to screen all of them. And that's where we discovered the Accutox 001, which is basically a variant of the Accume with 30-fold more potent activity on cancer cells. So now you can see that the IC50 went from 95 micromolar to 3.2 micromolar. So it became way more toxic against cancer cells. And the way to look at it is, for example, if you do an XN5 staining, so that's looking at apoptosis on T-cell lymphoma, colon cancer, melanoma, or breast cancer, you can see that the molecule is quite good, but in certain cases, the percentage of killing is not that efficient. So when we screen for the Accutox, here we used a concentration that is, so that's the second dose that was actually equivalent to what we tested here, and that's a lower dose that was still able to kill these tumors at a rate of 100%. And in fact, we did not only test it against those 4 different cell lines, we tested it against human cell lines, triple negative breast cancer, non-small cell lung carcinoma, colon cancer, melanoma, lymphoma. We've got about 11 of these different cell lines that were tested, and the outcome was always the same. This molecule is able to lead to 100% death over time. So the big question is, can we use it in cancer? So can we deliver it as an anti-cancer molecule in animals that have already an established tumor? And the answer is yes. This is an experiment that was actually done with the EL4 T-cell lymphoma, and that's an example we're showing you with one single immune checkpoint blocker. In fact, we tested it, the Accutox 001, with an anti-PD-1, with an anti-CTLA4, and with anti-CD47. And the CD47 is the one that led, again, to the best effect. This is why we think this is going to be the next generation best acting immune checkpoint blocker that I think Pfizer acquired lately from Trillium and is developing it into a phase 2, phase 3 trials. So if you look here, if you deliver, for example, the Accutox at 60 milligram per kilo, along with the CD47, which is shown here in red, the tumors are basically having troubles to grow. And the red arrows here represents the immune checkpoint blocker, CD47. The black arrows are representing the Accutox. So we only gave 6 doses at 48 hours between the doses, and the effect was actually astonishing. Now we know, in fact, that's something I didn't present here, but we know that the Accutox is also able to trigger what we call immunogenic cell death, meaning while you're killing the cancer cell is actually delivering signals to attract the immune cells, and it's also delivering antigens that the immune cells can actually capture to mount an immune response against it. So we're not only able to kill the cancer cell, but we're also mounting an immune response, which can eventually reprotect the patient or the subject from secondary tumors or tumor relapse over time. So these animals were 100% surviving, so 100% protection, and a very powerful control over time. So this is the T-cell lymphoma, but we also tested this technology against breast cancer, so using the E0771 model and the melanoma B16 tumor cells in this context as well. So we're also very active in the phase 1 trial. We would like to start a phase 1 trial here in Canada, where we're establishing right now all the documentations to approach the regulatory agencies, either the FDA or Health Canada, for a dose escalation study in combination with an immune checkpoint blocker. Of course, we said the PD-1, if we could have access to the CTLA-4 or CD47, that's something we're also looking forward to use. The sites that we are hoping to get our phase 1 trial is going to be multiple sites in McGill, the MUHC, and of course in Sherbrooke, the CHU of the city. So I hope I was able to actually give you a nice overview of the research activities we've been conducting so far in the company. Of course, we have way more data. We have data on CRISPR. We have data coming on the antibody drug conjugate program, which we believe are going to be amazing, because we could be applying not only that to antibodies or any ADC that, for example, has failed phase 1 trial or phase 2 trials or 3 trials because of efficacy or dosing, but we can also co-develop any ADC that is currently being in the program of a biotech or a pharmaceutical company. So this technology could be applied into DC vaccinations. So I showed you an example of melanoma, but all what you have to change is the lysate. So if you have lysate from breast cancer, colon cancer, glioblastoma, any kind of lysate axis, we can link the Accum to it, and then we can generate a DC vaccine against that indication. We've shown you that we can apply it to as a protein-based vaccine. So the example I gave you with HPV, but keep in mind that we also have done the same study with Spike 1. In fact, we've shown magnificent results with the COVID vaccine. The animals that were vaccinated, that was a study that was conducted, was a GLP study conducted at Vito in Saskatchewan, a level 3 lab, national level 3 lab in the country. And they were able to show that animals that were vaccinated against COVID have 0 inflammation in the lungs. So the lungs were clean. The nasal washes showed dying viral particles. The viral particles were not infectious. So we have 2 examples, and we're hoping eventually to drive more protein-based vaccines down the line. I've shown you that this molecule could be used as an anti-cancer molecule, as an Accutox. And of course, even though I didn't talk about that, the application that really fits very well with the Accutox technology is, again, the ADCs. And we're hoping to get data to present in the next International Investment Forum. So with this, thank you very much for your attention. If you have any further questions, please feel free to visit our website. You can have access to a variety of different press releases. You can send us an email to ask us about any information we wish to have access to, data room, confidential slide decks, and so on, if you sign an NDA, or you can call us and we'll be more than happy to discuss it with you. All right. So thank you very much for your time.
So, Dr. Rafi, thank you very much for the presentation. That was extremely interesting. We have 3 minutes left, and we have 2 very interesting questions. First one, let's say you have success with one vaccine and you adapt it against other tumors. What exactly do you need to do? What does your platform approach mean for adapting the vaccine? Are there advantage in regulatory process? For example, would there be such a process like that could change with new technology like yours, like a platform approval? So this is a lot of questions, but you have 2 minutes.
Yeah. So the answer is very simple. The vaccine itself, generating the DCs, is always going to be the same thing. So you extract DCs, you have them ready, and they're there. They're responsive to any antigen. If you have the lysate, which will be generated from a biopsy or after surgery, the chemical linking is always going to be the same. So if you use a lysate from breast cancer or melanoma or colon cancer, the process is exactly the same. It's just the protein lysate that is different, which makes it highly versatile to apply the technology to any kind of indication.
Perfect. And another question, how is the endosome being broken down before progression to the lysosome, or isn't the NLS at the inside of the endosome?
Yeah, so the NLS is part of the Accutox molecule. It's inside the endosome. The endosome is being broken through a ceramide production activity. So there's ceramide being produced, a chemical reaction, and it happens early on. So we're not waiting for the endosome to reach the lysosome stage. It's really happening early on once the endosome is at the early stage, which is an advantage, again, because it doesn't permit, for example, the acidification to occur and the proteases to be activated to destroy the cargo.
And lastly, we have about a minute to go. Obviously, maybe less pointed to you, but if you could tell possible investors, do you know if about your cash position, if you're going to be raising money soon, and so on?
That's an excellent question. I was hoping to get some to get that question from you, Julien. The answer is yes. We are basically expecting a lot of positive data in the next, I want to say, 60 days. And we are actually preparing ourselves to make a big raise for the 3 phase 1 trials we would like to start in 6 months from now. And in fact, we are building a very strong company. We're doing all the projects ourselves internally with combination, with CROs and sites for GLP studies. And one thing I'd like to add on is that we are actually preparing ourselves to go onto NASDAQ before the end of the year. So that's also gonna help us leverage.
Yeah, combined to your raise, I guess.
Exactly.
I think this is great, Dr. Rafi from Defense Therapeutics. This is always a pleasure to have you. Your projects are always moving forward very fast. And I don't remember many companies that provide such an exciting investment opportunities, especially that everyone knows someone basically that had one of the diseases that you're trying to treat, and the solution you're bringing to the table, or that could materialize, are absolutely mesmerizing and revolutionary. So thank you very much for being part again of the IIF. It was a pleasure to have you, and looking forward to the development at the next IIF.
Thank you very much, Julien, and thank you again for the committee for this opportunity. And I'm looking forward to have another presentation with you guys soon.
In October.
Right. Bye-bye.